Hi, everyone. My name is Swanson. I'm in the Department of Molecular Genetics and Microbiology, and it's my pleasure to introduce today's speaker, who was supposed to be me, but I managed to take a former graduate student, now a postdoc, and wrangle him into giving this presentation. There's a good reason for that, in that all of the work that you're going to hear, he did from start to finish. And he did this project not because he was encouraged to do this project, because trying to recover tissue, brain parenchyma from a stroke, can be considered a relatively high bar. But he decided to do this using an animal which has remarkable regenerative capacity. And sure enough, some of the aspects that we thought would happen did happen, but one of the results was completely unanticipated, and that will be towards the end of the talk. So let me introduce Ben Kidd, who's currently a postdoc in my lab.
Hello, everyone. All right, sounds good. As Maury said, I'm Ben, and today I have the pleasure of, he said he had to rope me into it. I'm happy to be here. But I'm going to talk to you today about spiny mice and the research I've been doing, looking at how they can recover their recovery profile post-stroke. So I'm going to give you a quick overview of today's talk. I'm going to give you a background on what spiny mice are, as Maury alluded to earlier, as well as start investigating stroke in these animals. Then we'll get into the results, looking at both the regenerative potential in spiny mice, as well as neuroplasticity post-injury. And then we'll finish up with some conclusions and future directions. So Maury mentioned this earlier, but spiny mice are a mammalian model of regeneration. So they're very different than a normal mouse that you see in the lab. They're from Africa, and they were actually initially discovered to have regenerative potential by Dr. Malcolm Madden in the Department of Biology here at UF. So they will essentially, in order to help escape predation, lose some of their skin. Snake bites them. Snake only gets a mouthful of skin. Spiny mice will run off, perfectly regenerate their skin with no scarring. And so that Malcolm discovered that. And since then, there's been a growing amount of research trying to understand, one, what leads to this regeneration, but what else can they regenerate? So that's led to different studies in skeletal muscle, kidney, ear, and I'm going to go a little bit more detail on the spinal cord. So when we actually were doing these experiments, there's another paper that was published looking at full transection of the spinal cord in spiny mice. And so this is some of the behavioral data from that paper. So on the left is a normal laboratory mouse. On the right is a spiny mouse. This is 8 weeks post full transection. And so what I hope you can appreciate is while spiny mice aren't back perfectly normal, they have much more robust control of their hind limbs than a typical laboratory mouse. So we're really excited about this. But this group went on ahead and actually went into quantify different behavioral recovery paradigms for these animals. So one of which is bladder control, which is a very standard way to look at after spinal cord injury. And so while normal mice never recover bladder control after a full transection, as seen in the gray dots at the bottom, spiny mice will recover by 3 weeks and be able to resume controlling their bladder. They also looked at what they call their BMS score. This is essentially looking at hind limb movement and digit extension, and kind of a really robust way to look at how much control they have over their hind limbs. And again, as you saw in the video, it's not back to normal. It's not perfect, but it is remarkably better than the spiny mice or the normal mouse. So kind of transitioning a little bit from the full transection injury, there was a group on campus here at UF that did a hemicrush. And so they went a little bit more into the histology of what happens. So this is in a normal mouse. In red is GFAP, or standing for astrocytes, looking at glial scarring. Green is NUN, standing for neuronal nuclei, and then DAPI for general nuclei. So if we zoom in on this, we see in a normal mouse, you have really robust astroglial scarring at the site of injury. They also stain for microglia/macrophages with IBA1, again, seeing pretty prominent microglial neuroinflammatory response at the site of injury in normal mice. That's standard. That's nothing surprising to anybody. But they wanted to see, what does this look like in a spiny mouse? So what they saw was a very diminished neuroinflammatory response, both with microglia and astrocytes. So with that, we were kind of getting really excited. It looks like they're able to behaviorally recover pretty well after spinal cord injury, as well as have a very much so diminished neuroinflammatory response. So we were wondering, can they regenerate their brains? A taller order than spinal cord injury for sure. But we wanted to go on ahead and try. And so when we were thinking about ways to investigate this, we really came upon stroke being one of the best ways, due to really the benefits it could have to patients if this worked. Really, the avenue forward would be remarkable, because it's really difficult to actually treat the damage. So if you could potentially treat after the damage occurs and regenerate the tissue, that's something that could be really cool. So today I'm going to be talking primarily on ischemic stroke. And as many of you probably know, ischemic stroke events are caused by blocking a blood vessel in the brain. So if you look where this red meets the blue, that is where the blood vessel is blocked by a clot, and then everything in blue downstream of that block is undergoing an ischemic event. So how do we diagnose this? In patients, we can do this non-invasively with CT and MRI. In rodents, we also will actually section through the brain and look at the damage we've done, cause them, as well as also looking MRI. So what does that actually look like in a patient? So this is a patient just a few hours after stroke onset. I'll highlight this section here. This is where the stroke is starting. And then when this patient came to a follow-up a few weeks later, the damage had been done and a lot of the brain was already dying off. So what's going on in the brain during this? So there's a really complex CNS response to ischemic stroke, one of which is going to be glial scarring. So you have those astrocytes being activated, walling off the damaged site, as well as neuroinflammation with macrophages and microglia. But in rodents, it's very well characterized that there is neurogenesis occurring, that rodents will try to send new neurons to this damaged site. However, it doesn't appear to be helping, at least to our knowledge as of right now. So what are the actual disabilities? What do patients experience? And this is incredibly variable. So this is going to be based on both the location of the damage as well as the size of the damage. In general, seeing some sort of partial paralysis and memory deficits are very common. In rodents, you have a lot more control based off of the type of stroke model you do. And typically what you see is circling. So the animals will perform circles after injury, as well as movement deficits and memory deficits. So how do we respond to this kind of insult to the brain? So there is compensation that goes on. The brain will actually rewire and reorganize how it works to help compensate. So really, in patients, this is limited and slow. There are neural connections changing. This is typically in both hemispheres, as well as the cerebellum. Rodents are a little bit, they're faster as well as limited, but they generally are compensating on the contralateral or the uninjured hemisphere. So what are our current treatment options? So for patients, the current treatments are tissue plasminogen activator, as well as mechanical thrombectomy, which are both fantastic if you are able to treat within the first few hours. However, if you're not within that really acute window, there's no really effective treatment outside of that. So because of that, and because of really the potential if we could regenerate tissue or have better functional plasticity, that's what I'm investigating today in the spiny mice. So I'm going to talk first about regeneration, and then I'm also going to talk about neuroplasticity, so how the brain could potentially be rewiring. But we'll start with regeneration, since this is a regenerative model. So we're able to induce strokes with the help of Eduardo Candelario and Pradeep Kamat. So this is done by opening up the neck for these animals, exposing the common carotid artery, and then inserting a silicone probe through the common carotid artery up to the base of the middle cerebral artery. From that, you then leave that probe in there for 1 hour, and that's blocking blood flow to everything downstream of the middle cerebral artery. So what does it actually look like in the animal? So this is a spiny mouse, just a few hours after we gave it a stroke injury and let it recover. So you'll see that classical circling behavior that we see in rodents after these kinds of injuries. So what I'm going to be showing a lot of today are MRI images. So this is actually a general 3D mask of a spiny mouse MRI, just to see. And what I'm going to show you is just a coronal slice towards the front of the brain. So this is what it looks like. This is what our MRIs look like on these animals. The contralateral throughout this entire talk will be on the left hemisphere. The ipsilateral, or the injured hemisphere, will always be on the right hemisphere throughout this talk. So I've outlined the injured hemisphere in red. You'll also notice, so that's indicated by this increased bold signal. You'll also notice very bright bold signals in the center of the brain. Those are the ventricles that are filled with cerebral spinal fluid. So you'll see those both on the uninjured and injured sides. Again, throughout this talk, the contralateral hemisphere will always be in blue circles, and the injured hemisphere will always be in red squares. So we can actually measure how much damage we're doing at 48 hours. What we found was that this was incredibly variable between individual animals. So these are very outbred animals. So we think that there's difference potentially in vascular wiring that's leading to this. But because of that, in order to actually see if these animals were getting better or worse over time, we needed to track the same animal longitudinally. So I'm going to show you a series of MRI sections now. So this is an uninjured spiny mouse. Again, you have those really nice ventricles lighting up on both sides of the brain. So the question is, all right, well, how does the brain look 1 month post-stroke? So what we see is that we have done a lot of damage to the animal and removed a lot of the brain over that time, and it's been filled with cerebral spinal fluid. So we can quantify this again. On the left is baseline, on the right is 1 month or 4 weeks post-injury. Again, the injured side is always going to be red squares. So this is great. Now the question is, can they regenerate their brains? Will this damage get worse, better, or will it stay the same? So looking at the same animal, we see that it gets worse. So again, we can quantify this and do see over time the damage is increasing. So this is really disappointing for us. And we wanted to go ahead and follow this up and just make sure that what we're seeing is in fact cerebral spinal fluid, and confirm that there's not underlying tissue there. And so there we go. So we did that and saw, yes, this is essentially now an enlarged ventricle in this brain filled with CSF. So when we're thinking about this failed regeneration in spiny mice in combination with the spinal cord injury, that we see that kind of regenerative response, we wanted to know, okay, well, what's going on differently? Is there, are some of the neurons being preserved? Is there any like neuroinflammation or astrocytes? So in order to assess that, I did multiple immunofluorescent stainings, staining for NUN for neuronal nuclei, as well as IBA1, staining for microglia and macrophages, and then DAPI. So we have our merged image here, and then we have some boxes there that I'll zoom in on the next slide. So what we can kind of see is that even at 2 days post-injury, that injured hemisphere is already showing a microglial response. If we can go ahead and zoom in, again, looking at NUN in the striatum now, and then IBA1 for those microglia. We're seeing that really increased microglial response in that area. So because we're damaging the striatum and the hippocampus the most in this injury model and the most consistently, I also wanted to look at the hippocampus at this time point. So again, going a little bit further into the brain, we see a little bit of microglial activation at 2 days, but we'll go ahead and zoom in on the dentate gyrus of the hippocampus. And we're seeing, again, an increase in microglial response at that time point. Okay, what about 30 days? So that's the very acute time point of 2 days. What's going on in the brain at 1 month? So again, looking at NUN and then microglia. And what I hope you can appreciate is a really strong localized neuroinflammatory response. In this animal, it was in the striatum and piriform cortex. So again, we can look in. And so what we found was that there were already starting to become lost neurons here compared to the uninjured side that line up really well with where the microglia are. So this is suggesting that the microglia or macrophages are phagocytosing the dead neurons and removing that tissue. So we can also, again, look at the hippocampus. We can see that we did a lot of damage to the hippocampus, so it's not really there on the injured side as much. And that really strong global neuroinflammatory response. So again, zooming in on the dentate gyrus, you see a really strong neuroinflammatory response with almost fragmented NUN staining. So it looks like they're really trying to phagocytose those dead neurons. So we can quantify this and again, see what I just described to you, that we are losing NUN positive cells in the dentate gyrus over time, as well as seeing an increased microglial response at 2 days that persists out to 30 days and out to 6 months in some animals. However, some animals did appear to have no more microglia activated at 6 months. So we're disappointed by that again, but we want to know, are they at least trying to mount a regeneration response? Are they at least trying? So in order to do that, we stain for SOX2, which when co-stained with GFAP, will label neural stem cells. So GFAP will also stain for astrocytes, so we kind of get to kill 2 birds with 1 stone with this and look at glial scarring. Additionally, we looked at double cortin for neuroblasts, so to see, is there more neurogenesis happening and are they trying to make new neurons to replenish the lost? So we're going to focus primarily on the subventricular zone, since that is one of the neurogenic niches in the adult mammal. So again, zooming in on subventricular zone at 2 days, we don't see much difference in either SOX2 or GFAP or double cortin. But again, we wanted to look at hippocampus. So we looked over at the hippocampus, which is another neurogenic niche in the adult brain. Looking at the dentate gyrus, we do see that there are neural stem cells present, as well as neurogenesis occurring on both sides. However, there is no noticeable shift at 2 days post-injury, which makes sense. The neurons aren't even gone at this point, so that tracks. We want to know long-term what's going on. So we looked at the 6-month-old time points, and what we see is much greater SOX2 and GFAP expression, as well as double cortin expression. So again, if we zoom in on that subventricular zone, we can see the astrocytes, while they are making a wall around it, there is a regenerative response, both with neural stem cell proliferation as well as neurogenesis. We also, again, looked at the hippocampus in these older time points. And again, that really robust glial scar around the damage. And then if we zoom in further, we can also even track these neuroblasts migrating to the damaged site. So they're at least trying to mount a response here. So again, we can quantify all this. So if we just look at astrogliosis to look at glial scarring, we see that it increases at the 1 month time point and persists to 6 months. We also looked at neural stem cell number, and we see that there is an increase in neural stem cell number at 6 months in both locations, and there is a difference at 1 month in the dentate gyrus. And then finally, we see an increased neurogenesis response at both the 1 month and 6 month time points. So there's no regeneration. So that was disappointing for us, but we really were thinking about the spinal cord injury and looking at that functional recovery that they got before there was really a regenerative response even. So we wanted to test that and see, okay, well, how are spiny mice recovering post-stroke functionally, not just anatomically? So we have a 66% survival rate for spiny mice in our hands. They typically will die within the first 2 weeks. However, generally, if they make it past 2 weeks, they survive. Similarly to the 2 weeks, we see that the vast majority of spiny mice will recover their weight within the first 2 weeks, back to 95% of their weight. There was one animal that took a lot longer to regain his weight. But while I was weighing these animals, I went in and I would check to see what we call neurocognitive deficits. So a baseline, they have a score of 0, which essentially means they don't have a deficit. They're running around, they're interacting just fine. A score of 1 is that circling behavior. So the circling behavior I showed you on that day of stroke, that's an N of 1, as well as any other kind of motor impairment that doesn't really paralyze them and they're still able to get food and water. But if there is some sort of motor impairment, we still record that as a score of 1. A score of 2 is if they have some sort of paralysis that's really stopping them from being able to get food or water, or if they're having seizures. So those are kind of the 2 hallmarks there. A 3 or 4 is essentially if they are dying or dead. So at the day of the stroke, at day 0, they're all circling. We see that they all have some sort of motor impairment. What I noticed is that the next day when I went to go check in on them, they were all running around in their cage, appearing to have normal motor behavior for the most part, not all of them. So I continued to go ahead and monitor this every day for the first 2 weeks and then each week after that. And what I hope you can appreciate from this is that while there are deficits, by that first week, they're all acting relatively normally. And if they make it to the first week, then they're acting normally past that. What this graph doesn't highlight is that a lot of the animals in yellow actually recovered and became blue. The animals that died were actually typically coming from the blue. So they're acting normally one day, I'd go in, and then I'd find them dead. It was very annoying. But so it seems that many of them are actually recovering from that. But we wanted to also go in and actually test this in a more kind of standardized test, because this is unfortunately kind of ambiguous. So we switched to including open field tests, since that's a very typical standard behavioral metric after stroke injury. We also did the corner test, but that one actually did not work because they learned how to crawl out of the corner, and they would just do that every time and not actually perform the corner test. What we did was we did these assessments at baseline, so before injury, 2 days after injury, 7 days, 14, and 28 days after injury. You'll notice that the number of animals is decreasing over time. Part of that is due to the fact that some of these animals died after the injury. The other part of it is that these animals also some of them learned how to escape the open field. So a very consistent behavior I would see is that they would jump against the wall trying to get out, and then the ones that learned that they could get out would consistently get out faster and faster each time. What we saw was we looked at total distance traveled, average speed, time and center, and percent turning clockwise, with percent turning clockwise being a surrogate for that circling behavior I mentioned previously. So they should, if they're circling, have a preference towards turning clockwise. What we saw was no changes. We didn't see any statistically significant changes. And so what that suggests to us is that they, by 2 days, were already, at least in open field metrics, able to kind of recover that behavior. Which makes sense when I think about the neurocognitive deficits. I was going in the following day and already seeing a lot of them kind of behaving normally. So we might just already, even at 2 days, be too slow to really see this deficit. So that got me thinking, how can we do a more non-invasive but acute monitoring of these animals' behavior? And so in order to do that, we recorded them for 24 hours, pretty much every day for the first week. So these are 2 male spiny mice in their home cage. They have not had any injury yet. So this is actually the night before we're going to give them a stroke. You'll see kind of classical chasing behaviors with them. You'll see them climbing up and down their cages. They're pretty fast, which makes quantifying this difficult. But what do they look like the night of the stroke? So these are the same 2 animals. The night of the stroke, you'll see that one animal is able to move around, get food and water. This other animal is actually in the corner. He's hard to see, but he did not move very much at all that evening. But even still, even the night of the stroke, you have that animal, which is actually recovering fairly well. Yes.
Evening being, this is the active phase.
This is the, yes. Yeah, so this is their active phase. So the videos I'm showing you are all at night. So this is when they should be more active. So if we look 24 hours later, these are the same 2 animals. See that they are a lot more mobile. They're interacting with each other more, appear to have more coordinated movements. So from here, we're kind of excited because, okay, we can actually see there is a deficit and they're recovering. So we want to go ahead and quantify this. So we did that with multiple metrics, one of them being movement. So what we did was we recorded the first 15 minutes of every hour for 24 hours for every animal. And so what you're seeing here graphed is actually the average number of events that happened within the first 15 minutes of every hour. In this situation, we're doing the night, so they're more active cycle. So each individual color is a different spiny mouse, so you can track your favorite spiny mouse over time. But what we saw was that there was a decrease in their general movement the first night of stroke, but that's recovered by the second night, labeled 1. And then that's persisted out to 1 week. And so what I'll also say is when we're doing our open field, we're doing it the day after this 1 time point. So this is the night in between, right before we do our open field. So we also looked at climbing, so how many times they moved up and down in their cage. And we do see a significant decrease the night of the stroke that is not significantly different out to a week, but also does appear to be slightly diminished. We also looked at circling at night. Again, we see that increase in circling at the night of the stroke, and then we see it decrease, and we see a very similar trend during the daytime as well. So we thought that's really cool. Oh, wow, that was loud, sorry. We thought that's really cool. Is there a way to actually assess this and what's going on in the brain of these animals to see what regions might be kind of compensating? So we're thinking about this and realize we have the tools to do this already in our belt. I showed you MRIs earlier in this talk. And so we decided to use functional MRI in order to investigate how the brain's connectome is changing after injury. So this is the magnet we're using. This is the 11 Tesla magnet in the Amherst facility right down the hall. So we're excited about this, but we ran into a hurdle in that we didn't have a spiny mouse brain atlas to be able to actually map to. And so in order to overcome that hurdle, I made a spiny mouse brain atlas. So the way I did this was I took 8 spiny mice brains, I perfused them with PFA, then PBS. We then left them in different fixative and then treated them with contrast enhancement for MRI. We then did high resolution MRI scans in the 11 Tesla magnet. From there, we took those 8 brains, we combined them together to make a composite spiny mouse brain atlas. And that's what I'm showing you here. So this is the composite brain going from the outside to the midline, the top to the bottom, and then the front to the back. So this is our high resolution brain scan. From here, I then mapped on 120 different brain regions, which are the standard brain regions used in these MRI studies, to our atlas. So now that we had our atlas, we have a way of scanning the mice. We wanted to know, does our atlas work? Is it up to snuff? Because one of the issues you run into with stroke research in this particular field is that the damage you do to the brain can sometimes distort and disrupt how those brains map to an atlas if your atlas isn't high quality enough. So to assess this, we took one of our injured brains and we mapped it to the atlas to see how well it does. So what you're seeing is our normal atlas is the brain you're seeing, and then our injured spiny mouse is a heat map on top of that. So the greater intensity in red is going to be greater bold signal, which is cerebral spinal fluid. So again, that's kind of the damage in the ventricles. And so what we're seeing is really nice mapping, both in the striatum and the hippocampus for this animal, and not being distorted anywhere else. So we have our atlas, we have our stroke model. Now we just need to go in, do the experiments, and see what's going on. So before I get into that data, I'm going to do a quick, brief description of how we analyze this. So this is an uninjured spiny mouse brain with its connectome. So what you're going to see are different, what we call nodes. Those are the yellow and the orange spheres that you see that are different sizes. And so a node is placed there, and that's where we're really measuring the activity. So those are the locations, those are the regions we're measuring. And those were chosen, 80 nodes total, 40 on each hemisphere, based off of general connectome, just getting an idea of the general brain, as well as our injury-specific regions. So from there, we can assess different things within the nodes, one of which is node strength. So this is demarcated by the size and the color of the node. So the more yellow and the larger the node is, that's the stronger it is. The more orange and small it is, the less strong it is. So what that's basically saying is, while we're doing our scan, how active is that node? And that's how we're getting that strength. Another phrase that you're going to see during this analysis is participation coefficient, which is less so focused on how much is it doing it, but also the timing of it with other nodes. So how active is really this node at the same time as these other nodes? How connected are they? And then finally, the last phrase you'll see is centrality, which in a nutshell is essentially the likelihood that that node is becoming a new hub of activity. So you can think of it like, is this a central node that is impacting a lot of other nodes? And that, again, is based off of strength and timing. So we have our baseline animal. What normally we'd see in a rodent model is that this contralateral hemisphere would be the one primarily compensating. So you'd see kind of this left side, the nodes would get bigger, and there'd be more connections. What we see after our injury is almost a distribution. A lot of nodes are actually decreasing, but other nodes that were less active are becoming more active. And so we're kind of seeing a bigger distribution of the activity in the connectome. So with that, when we look at how well the brain actually functions and is connected, when we look in stroke, both in patients and in rodents, the general connectome is decreasing. The strength of the connections are decreasing. And so we want to go ahead and just look at, in general, how is the brain connected? And what we saw was that there actually were no changes to our global network metrics. They were actually maintained 4 weeks after our injury, which we were really excited about. Because this kind of harkens back to the fact that they are behaving fairly normally. So with that, we thought to ourselves, okay, we see this global kind of stability. Are there regional shifts that are leading to that and contributing to that? So if we just start still a little big picture, but looking at the hemispheres as a whole, we see that we have that kind of contralateral shift, which is very typical in these kinds of studies. So that's that's not unexpected. We also see a trend towards the ipsilateral increasing as well. But we wanted to look at the regions that we know are getting hit. We want to look at the hippocampus and the striatum because we know they're getting injured. So what we saw with the hippocampus was that decrease on the injured hippocampus as far as its participation coefficient, so how connected it is with other networks outside of its immediate vicinity. And we're seeing a shift and an increase on the contralateral side. So what this would kind of suggest is contralateral compensation occurring, kind of what we typically think of going on in rodents. But we want to go ahead and look at the striatum because we know that's primarily getting injured. Again, we see that ipsilateral decrease, which is what we would expect, but we're not seeing any change in the contralateral side. So that got us thinking, okay, well, are there potentially nearby regions that are helping compensate for this damaged striatum? So we looked at the substantia nigra, and we did see an increase in the participation coefficient on the uninjured side. So again, this idea of like, it looks that, at least in these particular instances, the uninjured hemisphere is helping take over. So with that, we kind of got ourselves thinking, okay, it looks like it's not necessarily always a 1-to-1 ratio of hippocampus to hippocampus, striatum to striatum. So are there other regions that are very consistently changing? So with that, we found multiple regions that are likely becoming new neural hubs. So what that looks like, I'm going to go ahead and show you multiple graphs. In this particular situation, it's the posterior thalamic area. So we're seeing an increase in node strength, node degree score, participation coefficient, and node between centrality. So again, all these kind of harkening back to this idea that it's becoming more of a neural hub. So with all these regions, I'm also going to show you where those regions are in the brain, just to the right, to give it a little bit more context. So this, again, appears to be contralateral, the uninjured hemisphere. We also see the substantia nigra, again, coming on and most likely becoming a new neural hub contralaterally. We also see the amygdala, the medial amygdaloid region, both contralateral and a little ipsilateral. But also we saw the caudal insula really changing ipsilaterally. So we started to get some ipsilateral as well. And so that got us pretty interested. And the last one that I'm going to highlight to you is the flocculus of the cerebellum. So we saw both sides of the flocculus changing and looking like it's becoming more of a neural hub. So I thought this was really interesting because this isn't seen typically in rodent injuries, but it is seen in patients. So the cerebellum is a lot more involved in neuroplasticity in patients. It's thought to be because the cerebellum developmentally is really important for motor learning. And so the idea is you have those connections that were there during development that kind of get turned back on is one of the hypotheses in the field. So we think that also might be happening in these animals as well as they're relearning how to have that fine motor control. So these are the kind of the 5 regions that are really kind of dynamically changing. So in conclusion, spiny mice are not able to regenerate their brains out to 24 weeks or 6 months post-injury. They are making more neural stem cells. They're proliferating those more. They are making new neurons. However, it appears that they're failing to regenerate, presumably due to either neuroinflammation or astroglial scarring contributing to that. They are having rapid behavioral recovery, and there are regional changes in the connectome. However, to the global connectome, we're not seeing any significant shifts after injury. And there are those 5 regions that I mentioned that are likely becoming new neural hubs that are contributing to that. So future directions. I just talked to you about what we found so far in spiny mice. And one of the biggest future directions I have is really trying to understand what's going on in these brain regions. So one way to follow that up would be doing either transcriptomics, proteomics, metabolomics, trying to understand, now that we know what brain regions are changing, can we specifically target those and see what's going on in them that are different. Part of this can also be complemented by looking at different rodent models. If we're able to get a neural injury that's very consistent across rodent animals, we can kind of see what makes spiny mice different than other animals. So that's another avenue forward, as well as I mentioned to you a couple times of the spiny mice escaping different behavioral paradigms. So kind of optimizing and getting more robust behavioral paradigms for these animals would be really beneficial. Something I'm really interested in is trying to figure out what's going on with the glial scarring in comparison from the stroke injury to the spinal cord injury. And kind of, is that playing a role in the difference between the regenerative response there versus in our stroke model? Because if we can figure out that difference, that might have a lot of benefit to actually helping with the spinal cord injury and finding targets to really push forward there. Another thing I'm really interested in is what the microglia are doing. Microglia have been implicated in a lot of different facets of what I've shown you today, with neuroplasticity, phagocytosing the dead neurons, neuropreservation, keeping neurons alive. I'm really interested to see really what those cells are doing in these injuries. And what I didn't highlight today is that, but you may have noticed in the data, is there were 2 animals that didn't have any noticeable damage, but when I actually did histology on them, they had really robust microglial activation on the injured hemisphere. And so it's possible that for some reason those animals, either we didn't do as much damage, or the microglia for some reason were able to keep those neurons alive. So that's something I'm interested in looking at. Again, looking at variability in the vascular wiring that I mentioned previously, as well as neuropreservation, what's kind of contributing to this variation? And if we can kind of get rid of that variation, we can really start trying to figure that out. One way to do that is just trying to figure out, is there maybe a threshold? Maybe we just did too much damage to the brain. The spinal cord injury is a lot smaller of an injury, and so maybe it can recover that small injury, but if we just did too much, that's a possibility. One way to look at that is looking at alternative neural injury models and seeing, either with TBI or stab wounds, seeing, is there a smaller injury that they can recover from anatomically? So with that, I have a lot of people to thank. Like to thank everyone in the Swanson lab. Shout out to Dana, who is an amazing undergrad who helped me a lot with the MRIs and the behavior analysis. And this project would not have happened without the Malcolm Madden's lab, both Malcolm and Justin, phenomenal spiny mouse researchers, as well as Eduardo, Marcelo, and really all of my collaborators. This was a team effort for sure to get this done. So thank you for your funding. And I'd also like to thank the MBI and the CNG for helping fund me when I was a grad student, as well as the NIH and the Actimus Research Consortium on campus for their assistance. And with that, I'll take any questions.
No questions online right now? Can I get started? Okay, so this is kind of a two-part question. Do spiny mice develop tumors? Do you know that?
So there haven't been any natural tumors that people have seen develop in spiny mice. However, if you try to, hit them with a radiation, they will develop tumors. But they haven't, at least in bred in captivity, they haven't seen natural tumors, but they can develop them if you try to force it. Sorry? Possibly. Oh, I'm sorry. For everyone on Zoom, the question was, can spiny mice develop tumors?
The reason I ask is because you said frustrated and bummed out and all these things, because you don't see regeneration do you really want to see regeneration? I challenge that.
Yeah.
I know that regeneration is the appropriate outcome.
Mhm.
Regeneration that's not regulated may lead to, unregulated
Mhm.
cell differentiation, undifferentiated or dedifferentiation, or problem.
Yes, I fully agree that that would be a concern. Yeah.
You said you have reorganized.
Mhm.
Yeah. You also talk a lot about neuroinflammation,
Mhm.
gliosis, but you're only looking really at one or two markers.
Yes.
So, do you think that there is actual, like,